Hello and welcome to another episode of StratcomTalks. I'm your host, Jaffer Hasnan. Today we will be talking about innovations in biotechnology and how technology has changed the field of medicine. To discuss it with us, joining me today is Nick Bayless. He is the Chief Technology Officer at Centivax. Thank you very much, Nick, for joining us today on this podcast.
Jafar, thanks so much for having me. Happy to be here.
Now, Nick, to start off, tell us a bit more about yourself and your company.
Sure. I am an immunologist by training. I did my PhD in immunology, focusing on viral diseases and how viral infections cause disease in different folks in different conditions. I've been interested in vaccines for pretty much my entire scientific career. It's how I got my start in science and in biotech, was working for another vaccine developer years ago, and have always just been really passionate about the power of vaccines to affect human health, to eradicate diseases. To me, some of the most impactful stories in medicine and in biotechnology are the stories of vaccines and how we have eradicated entire viruses that were devastating to populations for the entire length of human history. Right. In the last century, we were able to eliminate or at least severely reduce a number of diseases, smallpox and polio and others. And when I was getting my start in science, I gravitated towards diseases that did not yet have vaccines for them. Influenza, we have a seasonal vaccine, but it's not super effective in normal years, and it doesn't protect us very well from emergent pandemic influenza like we've seen as recently as 2009. As well as other diseases that have never had an effective vaccine, like HIV, that has a number of challenges to getting an effective HIV vaccine, and one that can generate the right kind of immune memory in patients so that they are, protected from contracting HIV. That's been a persistent challenge in the field, and something I've been working on and curious to see move forward for most of my career. What Centivax does, the company I'm a co-founder of and chief technology officer of, we are a company founded to work on exactly these problems. We develop what we call broad spectrum vaccines and antibody therapies. When we say broad spectrum, I mean we're not looking to develop strain-specific vaccines the way the seasonal influenza vaccine works now, right? You have to get a shot every year because we reformulate it to match the strains that we think are going to circulate around the globe. And hopefully we do a good job with that prediction, and we can make a strain-specific vaccine quite quickly and get it distributed, around this time to folks all over the world. But it's a cumbersome process to have to be constantly doing that surveillance, making vaccines so rapidly and distributing them all over the world, and then only ending up with a vaccine that's partially protective against disease. It's not our best vaccine, and there's certainly room for improvement there. And so what we've done is we have developed a technology that lets us focus the immune response on parts of the virus that don't change from year to year. Lots of parts of the virus change in each annual strain that comes out, but there are small parts of the virus that don't change from year to year, or really even decade to decade. And we have a technology that lets us focus the immune response on exactly those parts.
Very interesting, Nick. I did not know that. Does it also apply to COVID-19, SARS-CoV-2?
That's a great question. When the COVID pandemic first started, we did an analysis and determined that, frankly, at the rate of mutations we were seeing for the first year and a half in the virus, we didn't think our technology was necessary. And we thought that the approaches being taken by Pfizer and Moderna and the other companies that have produced the vaccines would have been sufficient. And what I don't think anyone anticipated was the level of mutation that we were going to see come out of this coronavirus. We are now actively designing pan-coronavirus vaccine that will provide protection, ideally not just from all the COVID variants, but actually from the entire family of beta coronaviruses, which includes the original SARS virus, the MERS virus that emerged 7 or 8 years ago, and would apply the same technology we're bringing to influenza to the beta coronavirus family. Yeah.
Wow. And when is it likely to come out?
So to be honest, we had not started. We'd been monitoring, and we have an antibody medicine that we're making for SARS-CoV-2. So our other platform is working on that. But really, with the emergence of the Omicron variant and the highly mutated spike protein that the Omicron variant carries, that is where we started to say, you know what, I think our broad spectrum vaccine approach might actually be needed and necessary to get to the level of immunity that we want. And we no longer think that the traditional approaches are going to work. So it's this development process takes quite a while, but we're hoping to see some results next year.
I wish you all the very best with it. I hope it comes out as soon as possible. Now, Nick, vaccines have become a household name these days. Even as a journalist myself, if I'm not mistaken, almost every other day I read news on the television about vaccines. What I want to ask you here is, how has technology changed modern day vaccines?
It's a great question. So like we were talking about, there's been revolution after revolution in biotechnology and molecular biology over the last 3 decades. And a lot of those have moved the vaccine field forward in incremental ways. What we've really seen in the last year is the explosion of some of these very promising technologies. Number one on the list is mRNA. mRNA vaccines have been around in the HIV research field for a decade or two. I mean, people have been really interested in that. It was just the big advantage of mRNA is that you can respond rapidly to a novel pathogen, and that had always been the promise of mRNA technology. There had never been a great business case for developing and bringing an mRNA vaccine to market for an existing pathogen, because you would be trying to compete against the traditional flu vaccine manufacturers, for example, and they have had their process figured out for quite a while. This was exactly the kind of scenario that the mRNA folks have been talking about for a decade, that they would be best primed to respond to if a new pathogen came out and we needed to make a large amount of vaccine doses for a new pathogen. That's exactly where mRNA was going to shine. And it has been really exciting to watch and see Moderna and BioNTech working with Pfizer really take those technologies all the way through and have them be, some of the leading vaccines in the world. That is building on decades and decades of preclinical and early clinical development. So it's really exciting to see that finally reach market.
Indeed. And I have to say, their vaccines have served as a game changer in this coronavirus pandemic. Now, Nick, moving forward, certainly these innovations do not come without any risks, do they?
The mRNA technology is actually incredibly safe. And there is virtually no risk of any long-term or short-term side effect. It's an RNA construct, which is it's what we call unstable. It degrades fairly quickly. So when you get the mRNA vaccine, your body goes to work producing protein according to those RNA instructions, but the RNA instruction template itself actually gets degraded fairly quickly. There is no possible way it gets integrated into our DNA genome. There's no way it's staying around for very long at all. It's really a very, very safe and effective technology. We are actively talking to several companies in the RNA space. Our vaccine technology was originally developed using a more traditional protein-based approach. But of course, with all of the interest in RNA technology over the last year, we're actively exploring moving our platform over to the RNA.
Okay. Now, Nick, what kind of security protocols, safety protocols do you follow in order to determine that your vaccine is safe?
Sure. For our vaccine, we're currently conducting a number of animal studies in pigs and in ferrets. We will additionally conduct studies on other species of animals, possibly including some rats and hamsters, and possibly some small monkeys if needed. All of that data, we're looking with a sharp eye for any indication that there's any safety risk, any possible toxicity that would give us pause before going into humans. And then we will apply for permission with the FDA to start a human trial, and we will start in a very low dose, very safe way in healthy volunteers and increase our vaccine according to a predefined schedule that is vetted with the FDA to ensure absolute safety. We follow up on any patient in a trial for months or years, depending on the nature of the study. So certainly all of the large scale trials that Pfizer and Moderna have run, those patients in those trials will be followed for years to ensure and just make absolutely sure that there's no long-term side effects that we need to be worried about. But they're, like I said, these technologies are incredibly safe and effective, and it's great to see them come to market.
Indeed, they are. And I think we have already seen the results, right?
Absolutely. The one problem is that even with the RNA, it's still a very strain specific. I mean, we're seeing that now with reduced vaccine efficacy against the Delta strain and now against Omicron. And that's where the Centivax technology, where we have a way of creating a broad spectrum vaccine, our technology is designed to not see that drop in efficacy when the virus mutates, because we're targeting parts of the virus that don't change.
If I may interject, now, I'm no scientist, but as I understand, the vaccine you are making does not produce the spike protein, or it does not attack the spike protein. Is that how it works? It's still part of the protein cell of the virus.
Right. It still does direct the immune system to attack the spike protein, but it's like a hyper focused targeted attack on a very small part of the spike protein. Lots of the spike protein changes from variant to variant, and it makes a lot of little small mutations in the protein that knock out an antibody, because if an antibody was going to bind against an area that mutates, it's no longer able to bind, and therefore you're no longer immune to that strain. Our technology generates an antibody response against those parts of the virus that are the same from variant to variant and cannot change for various reasons of viral biology. And therefore, the immune response you get to the Centivax vaccines is going to be broadly protective against any strain that's out there and any variant or mutation that arises in the future.
This sounds so promising, Nick. Now, before I let you go, just one last time, summarize it for our listeners. How is your vaccine technology different from the one Pfizer and Moderna are using?
So the big innovation that Pfizer and Moderna have made is using an mRNA technology to deliver the vaccine, rather than traditionally we use proteins or whole virus as a vaccine. They're using an mRNA technology to deliver the instructions for making the spike protein. It actually gets made by your body's own cells, and then your immune system targets it. Our approach actually is, we're not currently using the mRNA technology. All of our experiments so far have been done on just protein, like many traditional vaccines have been done. What we do is we take a number of different variants of the virus and we mix them together and inject them at a low concentration, of a pretty small dose compared to what's out there in the commercial vaccines. And that allows the immune system to pick up only on making antibodies that are going to be completely cross-protective of all the different strains that are out there.
Wow. So promising, I have to say, and I wish you and your company all the very best with it. Just one last question, because this is the main theme of our discussion today. Since you are working in the industry of biotechnology, you have witnessed all this innovation yourself. You are working on one yourself as well. So tell me, how do you think technology is going to change the future of healthcare and different fields of medicine?
Sure. I mean, our ability to generate biological data has increased faster than the speed of computers or any other metric. We can sequence the human genome for less than $1,000 today. That was a $1 million proposition 10 years ago. The advances in sequencing costs, they've dropped to almost commodity prices. That allows us to do far more computational work leading into our experimental designs. And so each experiment we do is now driven by vast amounts of data that we didn't have access to even 5-10 years ago, that inform every experimental design and every vaccine design that we do. is absolutely all built on the back of how available sequencing data and genomic data has become. And I think you're going to see that all across healthcare with there's really incredible work being done on data in healthcare in terms of extracting information from patient records and doing large scale real world research, taking advantage of all the data that's now being stored in the cloud and in standardized medical record formats. Things like that are going to really accelerate personalized medicine even faster, I think, over the next 5 or 10 years.
All right, Nick Bayless, thank you very much for taking out the time for this podcast. I really appreciate your analysis.
Of course. Jafar, always good to talk to you. Thanks so much.